Hello, students. Please go ahead and pause the screen to complete your bell work. If you ever see a 0 in your bell work, that means that is non-responsive. You don't have to name the 3 organelles. You're thinking of them in your head. And then on number 1, it says name the 3 organelles that you're practicing today and their functions. I really want you guys to at least try to learn 3 per day, their function, and where they're located on your cell diagram from yesterday's practice bell work. Next, I'm going to take a second to introduce the new module. So your new module is the quadratic functions. There's a couple of ways you can view this. If you guys are using an app, there is a drop-down menu on the right-hand side of the screen. If you're using the computer, you can follow the same buttons that I just pressed. So to get here, I pressed home on the right side of the screen. Now we're going to scroll down. And this is the first place that you can see your modules or the announcements. And I like to use the modules button, or you can, again, like I said, use the navigation on the left side of the screen, which would be modules. And every day you also want to click on announcements to read the daily announcements, if there are any. Now remember, sometimes your bell work is within your lectures, like it is today. So this is where you have found the bell work. Anyway, let's click on our modules. We're going to scroll down. For this module, oops, wrong class, 1 second. Okay, now for this module, we have our Start Here page, but I'm going to be embedding videos into your unit module. So this is where this video will go when I'm finished recording it. So instead of having to click on these hyperlinks and viewing everything, you'll just be able to hit next down here to get to your next assignment. Now for your bell and exit tickets, like I said, they will either be in the announcements tab up here where you can click on announcements, or they will be throughout the lecture videos. So today's was in the lecture video. What you're going to want to do is you're going to click the assignment bell and exit for the week, and you can click to start the assignment, take the quiz, and then you would go down, since today is Wednesday, you would write for your bell work the 3 organelles that you chose to focus on today and their functions. Now you can submit this at the end of every day, or you can just ensure that it's saved by viewing right here. It'll tell you what time it's saved at, and you should be able to exit out of the questions and submit at the end of the week. This is due weekly on Fridays. If you're missing bell work for the first 2 weeks of class on Canvas, don't worry about it. I'm just counting those for exemption points, so you really don't have to worry. Plus, these are worth 4 points per submission, so a total of 4 times 8, which is 32, so you get 32 points a week for completing these, and they are easy points, just completion based, to make sure that you're practicing and staying on track to do well in the course. So this unit, we are talking about cells and cell functions, cell differences, and the organelles of cells. Cells were first discovered in 1665 by Robert Hooke. He was helping with some science studies and used a microscope to be able to first view cells. And then we had Anton van Leeuwenhoek in 1673, used pond water to see animalcules, which helped discover that cells have distinctive organelles, and that's where we know now that these organelles have distinctive functions. Here are the animalcules that Mr. van Leeuwenhoek saw in the pond. So these are more so like insects that we see today, but what he did see that was interesting was organelles, differences between plants and animals, which was very important to the current way that we view cells. So the theory of cells is that Cells are the basic building block of all living things. Organisms are composed of cells, and that cells come from pre-existing cells. So our new cells in our bodies, for example, are generated through the cell cycle, where we're able to go through replication of cells, cell division with mitosis and meiosis. Cells are very diverse and they vary in shape and size. If you've ever heard the saying that form follows function, that is very true for cells. So cells are in all different forms, and their forms usually have something special to do with their function. For example, we're going to start by talking about prokaryotic cells, which is part of the bacterial family. So bacteria are made from prokaryotic cells, and a lot of these prokaryotic cells have flagella or flagellum in the plural sense, and flagella are a whip-like structure that project off of the posterior side of these prokaryotic cells, and they have whip-like motions to help with locomotion. So form would be that it is a long tail-like structure, and its function is to help the prokaryotic cell move. So that is one example of form following function. Prokaryotic would be like, think about like having probiotics. Probiotics, like in your stomach, are going to help your bacteria grow in your gut, and prokaryotic cells have the example of bacteria as being a type of cell. Now, don't get confused. Viruses are not living things. They are not cells. So viruses are not an example of prokaryotic cells. So some interesting aspects of prokaryotic cells is that they do not have a nucleus. Instead, they have a sac or bundle of DNA within the nucleoid, which is the densely compact region of a prokaryotic cell, which is full of the DNA material. And the DNA of prokaryotic cells is in a circular shape instead of our double helical DNA structures that we see in humans. Some prokaryotic cells also have pili, which have basically the ability to anchor to other cells, and they function to help in asexual reproduction for these types of cells. And prokaryotic cells also can undergo division to reproduce. Examples of prokaryotic cells include Streptococcus and E. coli, which are both bacterial infections. Streptococcus is found naturally in all humans. It can be found in your nasal or oropharyngeal passageways, so it's basically a bacteria of the mouth or the nose. And Streptococcus, well, I guess I should say bacteria are usually named on their shape. So coccus is a description of the shape of the strep, and commonly you guys hear of strep throat. Strep throat is a common infection of your pharynx or your throat due to an overinvasion of Streptococcus, because too much of any bacteria or too much of any one thing can be bad. E. coli is another type of bacteria, and we hear about this frequently when we think of an outbreak in our produce. So E. coli is common to be found growing on plants, and if you don't wash your lettuce, well, you could get an E. coli infection. So make sure you're washing your lettuce. Other important organelles within a prokaryotic cell include the capsule, which is made of peptidoglycan, which if you think back to our macromolecules lecture, peptidoglycan is our protein macromolecule. So peptidoglycan capsule covers and encapsulates the prokaryotic cell, and it provides a structural and supportive and protective membrane for our prokaryotic cells. Prokaryotic cells also have a cell wall, a plasma membrane, which we also have a plasma membrane in eukaryotic cells. We have a jelly-like cytoplasm that helps basically suspend the different organelles in the three-dimensional shape, ribosomes, and plasmid in our prokaryotic cells. Okay, eukaryotic cells, if you're ever mixing these up, you have eukaryotic cells, and that is how I've always remembered it. So we as humans have eukaryotic cells, and it has DNA packaged within a membrane-bound nucleus. Examples of eukaryotic cells include plants, animals, fungi, and protists. Here is an amoeba, and it is consuming another protist. So you can see that little GIF right there of one of our eukaryotic friends engulfing another protist. Pretty cool. Okay, so the basic parts of all cells, and when we say parts, we're talking about organelles or important structures. All cells have ribosomes, and ribosomes are ribosomes have the function of synthesizing proteins. And if we remember back to our macromolecules chapter, proteins are made from amino acids, and ribosomes are able to decode the message from our messenger RNA to pair these base pairs with the corresponding amino acid, which is kind of interesting to think about. So ribosomes are hugely important to the functionality of all living things. Cytoplasm is the fluid or the jelly-like substance that is within the cell, and it forms the intracellular environment. So this is how our different organelles from eukaryotic cells are able to suspend their organelles along with prokaryotic cells. DNA is the genetic material of all cells. Cytoskeleton is the framework of a cell. It is the exterior framework of a cell. We've talked about chitin from our macromolecules chapter, which is an example of a cytoskeleton, which is going to encompass, for example, the exoskeleton of bees. And all cells also have a cell membrane, which is an outer barrier or an outer boundary that maintains two separate distinctive environments, the external and the internal cellular environment, also called the extracellular environment or extracellular fluid, and the intracellular environment or the intracellular fluid. Intracellular means within, intra, I-N-T-R-A means within. Now we are going to get into some of our organelles. This is not the cell diagram that will be on your exam, but rather the bell work where you practice and went online to practice for the labeling of the cell diagram is where these organelles will actually be pictured from. But we can see right here, ribosomes can be freely floating throughout the cell, but often they are found within the rough ER. They look like little tiny beads that stud or decorate our rough endoplasmic reticulum. And the function of ribosomes is to synthesize proteins. The nucleus is the brain or the power, or I would say it is the control center of a cell. It is surrounded by a nuclear membrane, sometimes called the nuclear envelope. The nucleolus, which is the densely packed center of the nucleus, is where ribosomes are synthesized. And DNA can be seen in the nucleus in a densely packed form called chromatin. And you can see that ribosomes are coming from the nucleus. Organelles are little organs of a cell, and they have all specific functions to carry out the overall bigger necessary functions of life. So we talked a lot about proteins in our last chapter, which is one of the main functions of ribosomes. And a lot of these different organelles have a function in protein synthesis. So when you're thinking of organelles, it might be very helpful to think of what do they help with protein synthesis. We have the endoplasmic reticulum, we have the smooth ER, and the rough ER. These endoplasmic reticulum act as an intercellular highway. So they are going to aid in our transport of different things throughout the cell. The rough ER is rough and studded with ribosomes, and the smooth ER has no ribosomes, so it's smooth. And so therefore, the rough ER is the site of protein synthesis, whereas ribosomes serve the function of synthesizing proteins. The Golgi apparatus, also called the Golgi body, is where the packaging and exportation of proteins occurs. So we go to the rough ER, where ribosomes are studded all over it. Here we will have the mRNA transfer its genetic material to the ribosomes, where it is decoded, translated into proteins. So these ribosomes are able to make these long polypeptide chains. Well, a long polypeptide chain isn't going to be able to travel as easily as if it were packaged up into a tiny, nice folded protein. So this is where the Golgi comes into play. The Golgi apparatus is going to take these proteins, package them, tie them with a ribbon, and send them off. How do we send these off? Well, there are vesicles that are formed in the Golgi, and these vesicles are where the proteins are packaged and being sent for export out of the cell. Lysosomes are digestive vesicles. So they look very similar to a vesicle that was made in the Golgi apparatus, but these lysosomes are having different functions than to carry and transport proteins. Rather, they are going to have digestive enzymes that are going to break things down, and they are also known as the suicide sac. So LYS, this is a medical term meaning to burst or break. So lysosomes are enzymes, or I should say are vesicles that contain enzymes that will break things down. It's basically the trash can. So we will have things we don't need anymore put into a lysosome, digested, and then sent out of the cell. Babies with Tay-Sachs disease have defective lysosomes. Because they cannot break down these waste products, they build up in the cell and cause major brain damage. Babies with Tay-Sachs die in early childhood. We will have an assignment on Gaucher disease that also coincides with this chapter upcoming. Lysosomes are also called the suicide sac because a cell that has become damaged can destroy itself. This is called cell lysis or apoptosis. And it does this by releasing the contents within the lysosomes to have a programmed self-destruction. This is not by accident. This might be because there was an issue with the cell functioning, or maybe it had become infected with the virus. Who knows why this is happening? But anyway, this is one process that can occur, and it occurs through the process of lysosomes. Okay, here's your exit ticket for the day. If you have any questions, please feel free to reach out to me. Have a great rest of your day, and I look forward to seeing you all soon.